okay, so that you can get the information. Hello and welcome all students to another video tutorial from Genome, and I would be your host, your faculty, Dr. Nitin Wahi, teaching you about the basics of amino acids and protein. So this constitutes the first basic and the foremost chapter in the field of amino acid, protein, and protein biology. Now, basically, what are amino acids in nature? So amino acids are just linear unbranched polymers constituted by the 20 standard alpha amino acids. Now, when you look towards amino acids, there are actually four fundamental biomolecules present in the world. Which are the four fundamental biomolecules which are present? One is referred to as amino acid and proteins. The second one is referred to as lipids. The third one is referred to as carbohydrate, and the fourth one is referred to as nucleic acids. Okay, so we would be initiating our lecture with amino acids and protein. I have already told you what is nucleic acid, their structural and functional component. Now we would be talking about the most versatile, the most enormously diversified component of the biomolecule, and that is referred to as amino acid and proteins. So we'll be talking about all the different types of amino acids and protein that constitute the structural framework of our body. Okay, so proteins, basically what are proteins? Proteins are linear unbranched polymers of 22 alpha standard amino acid residue. They are encoded in the DNA of the genome. So what is going to happen? So proteins molecules, okay, there is some disturbance, Rishla. I didn't recognize that up. Okay, just muting all. Okay, now please look here. See, proteins are linear unbranched polymers consisting of 22 standard alpha amino acids that are encoded within the DNA of the genome. The fundamental unit of protein polymers are the alpha amino acids. Now, very important point. You got two very important points in this particular unit. See, I won't have any animation today, but look at my words and the things that I'm going to highlight, because that will form the basis of all the studies of biochemistry that you would be doing sooner or later. So basically, proteins are linear unbranched polymers constituted by 22 alpha standard amino acids. Okay, here there are two words which are very important. The word linear here is very important, and the word unbranched is also very important. I'm just closing it up. These two words: linear unbranched polymers consisting of 22 standard alpha amino acids. So if you're talking that there is a protein molecule, and this protein consists of linear unbranched polymer molecules present within itself, then how this protein is going to stabilize its structure? Because it is unbranched, it is an unbranched molecule, okay, and then there is a linear molecule. So supposingly if there is a linear molecule and there is an unbranched molecule, if there is a molecule, supposingly this is protein, okay, this is a protein, okay, now this is a protein which is an unbranched structure, okay, now this is also a protein which is also an unbranched structure. Then how these two proteins are going to interact together? What would be the mechanism of interaction in between the two different proteins? Those proteins, how these two proteins are going to interact with each other? Because see, proteins can never have cross bridges. They are linear, they are unbranched polymers. Proteins are linear, proteins are unbranched polymer. As these proteins are linear and unbranched polymers, so how they are going to link together with each other? Okay, so what we have to decide here is, how does these proteins interact with each other? The proteins are going to interact with each other via the formation of disulfide linkages, the cross bridges that are going to be produced within the structure of the proteins, or via non-covalent interaction in the form of other different non-covalent interactions, such as hydrogen bonding, hydrophobic base, hydrogen bonding, ionic interactions, salt bridges, weak van der Waals forces. So those will be the interactions by which the proteins are going to interact with each other. See, there are two proteins. Now you look at your own hand. Okay, you have your own hand. I'm just switching on the video. Look at the video. Look at my hand. See, if this is a hand, and there are proteins constituting the structure of my hand. That means what I'm saying here is there is a protein that is referred to as collagen. Okay, that protein is going to constitute the structure of my hand, and every protein is linear and unbranched as told to you in the definition. Then if I'm going to bind a fist, then what is going to happen? The proteins are going to get separated and the bones will be reflected out. The blood will ooze out. Why it is not being oozed out? While the proteins are linear and unbranched structure, what keeps the protein intertangled? The proteins are being kept intertangled with the help of disulfide linkages. Okay, proteins are being held with the help of disulfide linkages in cross links. Okay, they form linear structures, but those linear structure also binds to each other by disulfide linkage. Suppose this is a disulfide linkage, this is one protein, this is another protein, so they are going to form a stabilized structure. Okay, next thing, the two proteins which are together with each other in a linear unbranched manner will interact with each other through hydrogen bond, through hydrophobic interactions, okay, through salt bridges, through van der Waal interaction. Now, what are these interactions? We would be looking forward in detail, in great detail, in our theory. Okay, I do think that my voice would be hearable. If it is not, please just inform me up. Vandana, Raj, you can inform me up so that I can get it up that my voice is today audible to you all or not, please. Okay, now the next point that we are going to study here is, okay, it is audible. Okay, thank you. Amino acids are composed of an amino group, a carboxyl group, and an hydrogen atom present within their structure. Okay, when you look towards the structure of amino acid, now we will be looking at the structure of amino acid. What is the structure of amino acid and why they are such an important compound in nature? The structure of amino acid, as you can see, is given in the figure. See this, amino acid consists of a chiral carbon atom which is being bound to a carboxylic group, amino group, a side chain, and a hydrogen atom. Okay, usually this carbon is chiral. So amino acid is actually composed of an amino group, a carboxylic group, a hydrogen atom, and a side chain present within its structure. And the centralized carbon atom to which all the different molecules are being bounded is referred to as the alpha carbon atom. Okay, so this is the alpha carbon atom, and there are groups which are being bounded to it. Okay, now the most important things, that how these amino acids form a complex structure, that of protein. The structure is common to all, but there is one exceptional amino acid residue which do not follow the classical definition of the amino acids. That means the carbon there is not chiral in nature. That amino acid is referred to as glycine. Okay, so this structure is common to all, but one of the alpha amino acid that has a cyclic structure is also an exception, and that is referred to as proline. The R group, the side chain in the case of alpha group, alpha carbon is different in each amino acid. I will tell you the tricks how to remember each structure, and then you can effectively memorize all the different structures of the diverse amino acid molecules. We would be looking at it. Okay, in the simplest case, the side group can be an hydrogen atom, and this is the structure of glycine. Okay, now the questions which have been asked in your examinations are from the first note that you must underline. Now these are some of the questions which have been asked in CSIR and other different entrance examination. Now, which are those questions that you must understand? The first question that I'm going to inform you of is about certain facts related to the amino acids. First fact, glycine. Glycine is the simplest, most conserved amino acid residue. It has a minimum molecular weight of 75 Dalton. Dear all students, these facts have to be memorized. You have to learn them by heart, because questions have been asked in several different entrance examination. बहुत सारों में questions पूछे गए. Many times it has been asked. Okay. Glycine is the simplest, most conserved amino acid. It has the minimum molecular weight that is possible. Okay. Now, tryptophan. It is the heaviest among all the amino acids known. It has a molecular weight of 204 Dalton, and it has the least occurrence in protein. That means only 1% of the 1.4%, less than 1.4% of the protein is made up of tryptophan. Okay. The most acidic amino acid. The most acidic amino acid is aspartate. It has an isoelectric point of 2.77. You have to learn this by heart. The most basic amino acid is arginine, which has a maximum isoelectric point of 10.7. Okay. Most hydrophobic is arginine. Arginine was also the first discovered amino acid in nature. Okay. Most hydrophobic, the most, oh sorry, this is hydrophilic, sorry, this is the most hydrophilic, arginine. Next is most hydrophobic amino acid. The most hydrophobic amino acid is isoleucine, ILE. Okay, this is isoleucine. Okay, and the amino acid with maximum number of rotatable bond is leucine. Amino acid with maximum number of rotatable bond. Amino acid with maximum number of rotatable bonds is leucine. Okay, and this is the amino acid which is found maximally in protein. And this amino acid is approximately 9.1% of the total protein concentration which is being found. Okay, so these are some of the facts that have to be memorized, because these facts have been asked in numerous times in different competitive examination, including CSIR, Section B, ICMR, DBT, and in other entrance examination. New students, if you are there, Amu, you are there, Akhilesh, you are there, and there are some other students who are new in the batch. If you find any difficulty, you can always type your query, and your query will be answered in the class by itself. Okay, so this is something that you have to memorize. The recording of the lecture will also be provided to you after the class. Okay, now moving forward, you see the structure of lysine. There I have written something. Can you recall what I have written here? See the maximum number of rotatable bonds which were present. Okay, that one was present in the case of leucine. Okay, no problem. Now see, look here. This is a basic amino acid residue. This is a basic amino acid residue because in the side chain there is an amino group which is present. That is why it is basic. Now see how the carbon atoms are being numbered, or they are being labeled. The first carbon atom is being numbered as alpha, and subsequently the second carbon atom would be beta, third would be gamma, fourth would be delta, fifth would be epsilon naught. So there are carbon atoms which are being labeled as alpha, beta, gamma, delta, epsilon naught, and so on. So in alpha amino acid residue, the number of carbons are being labeled as such. Now the first properties of alpha amino acid. There are certain properties of alpha amino acid which we must remember. The first property of amino acid is that they behave as zwitterine. That means they can behave as an acid as well as base. As these can behave as an acid as well as base, so they have an amphoteric nature. So these are referred to as amphoteric molecules. Okay, as these are referred to as amphoteric molecules, so they at lower pH will will absorb the proton, and at higher pH will donate their protons. Okay, so how does the reaction takes place? See, at high concentration of the hydrogen ion, that means at low pH, the carboxyl group accept a proton and molecule become uncharged. Okay, So what is going to happen? Amino acid also act as a buffering agent. Do you know what is buffer? A buffer is a compound that resists change in its pH. Any compound that resists change in its pH is referred to as buffer. If a change is going to take place, then that compound is going to oppose that change. Okay, then it is referred to as a buffering molecule. Okay, so at high concentration, when there is low pH, the carbonyl group will accept a proton and it will try to maintain the pH. Okay, now what happens when similarly at lower concentration of hydrogen ion, when there is high pH, what is going to happen? When there would be high pH, the amino acid loses its proton and become uncharged. The amino acid will lose its proton and it will become uncharged. As it becomes uncharged, so it will cause the decrease in the pH to take place. So amino acids are the one that will oppose any change in the pH of the solution. Okay, so now how does amino acids are how does this isoelectric point of an amino acid is being tabulated, and what is this isoelectric point? What is this pi, the most important property of the amino acid molecule? What is this pi, isoelectric point? Now see this, the point at which amino acid occurs in the form of a zwitterine. It has a positive charge as well as it has a negative charge at that point, and the sum total of all the charges over the amino acid is equal to 0. Then it will behave as a zwitterine. Now, the pH at which the amino acid behaves as a zwitterine is referred to as the isoelectric point or isoelectric pH. Now, there are two different types of pH, two different types of reactions that we are going to visualize. Now, what we are going to do here is that we are going to titrate the amino acid. Now, this is an amino acid. Now, we are going to titrate it out in a titration reaction. Now, what we are doing in the case of a titration reaction, I'm just opening a whiteboard in front of you, that what we are doing in the titration reaction. Okay, probably this board will be clear to you. Okay, now in the case of titration reaction, what we are going to do here is that we are going to take a tube. Okay, this is a burette that we are going to take, and from this burette, what is going to happen? We are going to control the movement of molecules. Okay, how? We are drawing an arrow. This arrow indicates that we are going to control the movement of molecules, and the molecules are going to fall in a flask. These molecules are going to fall in a flask. Okay, there is a flask that I'm going to make. Probably I will. Okay, this is a flask. Supposingly, this is a flask in which the molecules will be made to fall. Probably I will be able to make. Now, what is going to happen here is that I'm just making a flask. Just a second. Just a second. Give some time to me. Okay. Probably this is a flask that we have made, and now you're titrating, and you have added some acid here. You are titrating that acid with amino acid solution. Then amino acids are going to fall into that acid. As amino acids are going to fall into the acidic solution, we are going to see that at what pH the the amino functional group will gain a proton. At what pH the carboxylic functional group will gain a proton, that will be referred to as the lowest pH or the pKa. Now, what is pKa? Let us move to the Pathfinder to understand it up. Now, see this. Now, when titration is going on, firstly, what will happen? Firstly, there would be an acidic environment. Now, there is an acidic environment. Okay. Now, into this acidic solution, you are also going to add base. Now, what you are going to do here is that, sir, class, hello, class. See, what we are going to do in this particular reaction, see, please look at the annotator, there was some disturbance. Now, what is going to be done here is that the first amino acid within the solution will be present. I'm just labeling it out. If I could label it out. Okay, I'm just trying to label it out. There is amino acid in acidic solution. Okay, this is acidic solution. Okay, so the pH, hence the pH is low, hence there is a very low pH. PH is lower than 7. Okay, do you get to understand that? Now this is the amino acid in positive in acidic solution. It is titrated with a basic solution or base. Now base is being added into it through this burette. A base is being added into this amino acid acidic solution. Then what is going to happen? Again, moving to, then the pH is going to be increased. When you are going to add base, the pH is going to be increased. The pH at which the carboxylic functional group loses its proton is referred to as pKa. Now please do note it down. I'm just going to write it down here. See this is the functional group that we are looking forward. Okay, now what we are looking forward is that during titration reaction, just during titration reaction, the pH at which the carboxylic functional group, the COOH group, loses its hydrogen, its H positive ion, is referred to as pKa. Okay, this is referred to as the pKa. The pH at which the carboxylic group is going to lose its proton is referred to as the pKa. You find that this is the point where the pKa lies. Okay, now again you are going to add more amount of base. Then what is going to happen? At certain level, there would be no charge on the amino acid residue. Now again and again you are going to add base. Then the pH is going to be greater than the isoelectric point. When the pH is greater than the isoelectric point, then the positively charged amino group will also lose its proton at a pH. Then that pH is referred to as pKb. Now this one was the first pKa. Okay, on further reaction, what is going to happen? There is another molecule. On further reaction, what is going to happen, that we have to see. On further reaction, further addition of a base, the pH further increases above pi. Okay, it further increases above pi, and so the pH at which NH3 positive, I cannot write 3 there, NH3 positive loses its proton, is called as the pKb. It is referred to as loses its proton is referred to as pKb. Which group? NH3 positive group loses its proton, then it is referred to as pKb. I'm just minimizing this up so that you can clearly visualize it out. Okay, here now it would be fine. I'm just placing it at the right place. Okay, now it would be fine. On further addition of a base, the pH further increases above pi, and so when the pH at which this particular amino group is going to lose its proton, then that pH will be referred to as pKb. So this reaction is taking place at pKa, this reaction is taking place at pKb. So what would be the isoelectric point? The isoelectric point will be present in between pKa and pKb. So we get a formula, and that is, for a normal amino acid, the isoelectric point calculation is pKa plus pKb, or pK1 plus pK2 divided by 2. Okay, I'm just labeling this out here also. This one was the second. This one was the pKb, which was being hydrolyzed. Okay, in this particular, which was being broken down to liberate the proton. Okay, now this is what we are going to get in the reaction pathway. Now, the simple questions that are being asked in CSIR entrance examination is something like this that you can find in the MCQ booklet also, where you are going to find that how the reaction is going to be proceeded. In that booklet, you are going to find that, for example, for an amino acid residue, if resi, if pKa is 4 while pKb is 10, the pi is the pi is, you just have to calculate it with the formula pi equal to, sorry, pi equal to pKa plus pKb divided by 2. pKa plus pkb divided by 2. Okay, so you get that particular answer, that is equals to 7. Okay, just calculate it out. 10 plus 4 divided by 2, 10 plus 4, 14 divided by 2 equals to 7. Such questions have been asked in section B of CSIR net entrance examination. Such an easy question, but now they are outdated questions. Now such questions will not be asked. Now certain other modifications have been made that we have to look forward. Now, which are those modifications which have been made? Now, this is an old pathfinder that you are visualizing here. In the new pathfinder, this hydrolysis is being given for the acidic as well as for the basic amino acid residue. So let me check if it is given, then I can have the question being answered here only, else I will make the scanning of the new pathfinder and will show it to you. Now look here, these are the questions which have been asked about the isoelectric point. If we can find it out, let me see if you can just recollect. Now see this, the first reaction that I was telling to you, I was informing you the first property that amino acid can act as a zwitterine. The question is directly being asked from this titration of amino acid. You are going to titrate an amino acid during titration with a strong base such as NaOH, alanine. Supposingly we have taken alanine, my dear friends, then what is going to happen? Alanine is going to lose 2 protons in a step-by-step manner. There are 2 protons which are going to be lost in a step-by-step manner. Now, can you recollect where from these 2 protons have been lost? The first proton has been lost from the carboxyl group and the second proton from the amino group. The pH at which the carboxyl group was losing its proton was called as pKa. The pH at which the amino group was losing its proton was pKb. Okay, so what is the formula? You can look forward here. See this, at low pH amino acid has +1 charge. Another question which was asked in CSIR back in 2012, okay, in section B. At high pH amino acid has -1 charge. Another question which has been asked. The point at which the amino acid has no charge is called as isoelectric point, or the pi. Now how this pi can be calculated? Pi can be calculated as the midpoint in between the pK2 and pK1. So you just add pK1 plus pK2 and divide by 2, you will get the isoelectric point, that is referred to as the pi. You will get this particular point, okay, that is referred to as the isoelectric point, that is pi. Now please listen to me, very important. Now see, you can find there is a whole range of variation that are present here. Now which is that range of variation? I would like to point it out. You are finding this particular range. I'm informing you about this particular range. Right from pK1 to pK2, there is a range that is present. In this entire range, the amino acid will be present in the form of a zwitterine. Okay, this amino acid will be present in the form of a zwitterine. In this particular range, from pK1 to pK2, the amino acid will be present in the form of a zwitterine. Here the amino acid is present in the form of zwitterine. In between the pH variations of pKa1, pKa, and pKb, pKb amino acid exist in the form of a zwitterine, and this variation is referred to as isoelectric range. Okay, and this pH, which is greater than pKa and is less than pK2 is referred to as referred to as isoelectric range. Okay, this is the isoelectric range that you're going to find. Okay, is this clear to you? You can note it down in your book, in your notebook, that this is the one where the pH is present from the pK1 to pK2. There is a range which is present, and that is called as isoelectric range. Now, questions have been asked, which molecule has which isoelectric range? I've told you a formula on the first page. See this? This is that formula that is being shown here. I'm just moving away. Okay. This is the formula that is being shown to you. I will have to delete this up, else this won't move. Sorry for that. Save. Okay. Now, can I move it? Probably no, I can't. Just a second. Okay. Now I'm moving. You will get the same formula here. Now, what is the formula that is being shown here is pK1 plus pK2 divided by 2. So this is, so supposingly there is an amino acid which has 2 pK's value, pK1 being 2.34 and pK2 being 9.7. Then the calculations comes out to be equal to 6. You can look at the MCQ booklet. First 5 questions are on this concept only. Okay, so this is a very easy concept. Now look at another amino acid. Now we are going to look at an amino acid that is acidic. Okay, and then we would be looking at another amino acid that would be basic. Okay, they are not showing it for the basic one, but they are showing it for the acidic one. In the new edition, the book that you all have, you have all taken the book from me only. So that book is having this carboxylic functional group as well as the example of the amino functional group. Now see, when you're talking about an acidic amino acid, in the case of an acidic amino acid, there are not only 2 groups. There is another group, that is another side chain carboxylic functional group that is present. One is the main chain carboxylic group, another is the main chain amino group, and there is the third group that is present, that is the carboxylic group which is present. The pH at which the main chain carboxylic group is going to be ionized is referred to as pK1. Okay, also referred to as pKa. Okay, so see this, what I'm going to tell to you, I'm going to inform you of the most important trick. The most important thing to be remembered here is this, whether the amino acid is simple or whether it is acidic amino acid. In the case of acidic amino acid, carboxylic functional group of the main chain is ionized first, and in the case of basic amino acid, the side chain amino group will be ionized first. Okay, so this has to be remembered. You must remember this out. Okay, now what we have to study is that after the ionization through this first point where the pKa is 2.19, what we get, that there is an amino acid that has been ionized. So this has reached an isoelectric point. Okay, because this has become a zwitterine. Now when the side chain is being hydrolyzed again, then it will get converted into 2 negative charge and the zwitterine condition is being lost. So where is the zwitterine? Where is the isoelectric point? So the isoelectric point is present in between pKa and pKr, or in between pKa or pK1 and pKr. So the formula for an acidic amino acid, would be pKa plus pKr divided by 2. divided by 2. Now see, I'm just writing it out here to explain it much more clearly to you. In case of, note, this is the most important point. In case of an acidic amino acid, the carboxylic functional group of the main chain is ionized firstly. Okay. Now, why it has been ionized firstly? Because this is a carboxylic group. This is a carboxylic group. Carboxylic group means it is acidic. अरे भाई carboxylic group का मतलब क्या? Acidic. So carboxylic group means acidic, and that is why this is being ionized firstly. Now see, the pH is less than 7 only. Then this group will also be ionized. So this has also been ionized, and here you get the formula for acidic amino acid. For acidic amino acids, you get the formula for isoelectric point, Pi equal to, what would be the formula? pKa plus pKr divided by 2. Okay, this would be the formula for the acidic amino acid residue. Okay, for acidic amino acid. Now for basic amino acid, I'm just going to show you the formula here only. Okay, so that you can compare the different formulas together with each other. Okay, for basic amino acid, this thing is going to be altered. That is given in your books. You can open your book, you can look into it. The formula for calculation of isoelectric pointer Pi will be equal to pKb plus pKr divided by 2. pKb plus pKr divided by 2. Now what is pKa? What is pKb? pKa, the point or the isoelectric point at which the carboxylic group has ionized. Okay, pKb, the pH at which the amino group has ionized. Okay, and for a normal amino acid, normal amino acid, the formula becomes equal to Pi equal to pKa plus pKb divided by 2. That is pKa plus pKb divided by 2. Okay, this becomes the normal formula for the amino acid residue. Now the note is that in case of acidic amino acid, the carboxylic group is ionized first. Carboxylic group of the main chain is ionized firstly, while in case of a basic amino acid residue, the amino group is ionized at last. Now there are more than 10 questions given in your Pathfinder MCQ as well as in test and evaluations, which is based upon these formulae. Okay, for acidic amino acids, simply you just calculate pKa plus pKr divided by 2. For basic amino acid, you just calculate pKb plus pKr divided by 2. And for a normal amino acid, it is very simple, pKa plus pKb divided by 2. pKa sometimes is also written as pK1, and sometimes pKb is also written as pK2. pKr is always written as pKr. So that's all. In this manner, you can calculate the isoelectric point for a different amino acid, whether it is acidic, whether it is basic, whether it is a normal amino acid in nature. Okay, so majority of this thing will be clear to you, that how to calculate these all amino acid residue. There are questions given in Pathfinder. You can look at those questions, and if you cannot solve, I will help you in solving them out. Okay, we will discuss the questions in our group. Okay, after the class. Now see this. This is how the reaction takes place. Now in this reaction, what you are going to find? In this reaction, there would be 3 different peaks. Now see this. There are two different peaks that you can find in this particular reaction, but here there would be pk1, pkr, pk2. So there you will get pk1, pkr, pk2, and the isoelectric point will be present in between pk1 and pkr. That means here there would be an isoelectric point for this acidic amino acid. Okay, and in the case of basic amino acid, what is going to happen? In the case of basic amino acid, there would be again three pH, pk1, pk2, pkr, and the isoelectric point will be present in between pk2 and pkr. Okay, that is why the formula is for basic pkb or pk2 plus pkr divided by 2. These are the formulas that has to be applied while solving the questions. Please try to attempt those questions which are given in Pathfinder. Now I'm just asking you to calculate. You can take your copy and pen. You just take your copy and pen and try to calculate the isoelectric point for these different types of amino acid and compare it with the information chart which is given in Pathfinder. Okay. Now this is the pk1, pk2. The every question pk1 and pk2 will be given to you. Then only the calculations can be made. Now moving backward, the first property of the amino acid according to me is over. Okay. Now, just try to solve all these questions. Some of the questions are very important for leucine and for tryptophan. Now, we are moving backward to the page number 1 of Pathfinder, where page number 2 that we were talking about. Now, after this, there is another property of the amino acid residue. What is the next property of the amino acid residue? All amino acid residue are optically active except glycine, because they contain a chiral carbon atom present within itself. Okay, because they contain a chiral carbon atom present within itself, so these amino acids always have the ability to rotate the plane of polarized light either towards right, then it is referred to as D amino acid residue, or towards left, then it is referred to as L amino acid residue. So amino acid residue can be dextrorotatory as well as it could be levorotatory in nature. Okay. Now moving forward, see, a quantitative measure of the optical activity of a molecule is called as optical rotation. When you measure the optical activity of a molecule, when you will try to quantify the optical activity of a molecule, then the phenomena is referred to as optical rotation. You are measuring the amount of optical activity that a molecule possesses. If you are able to measure the amount of activity of the molecule, then it is referred to as optical rotation, and optical rotation is being given by the term alpha. Okay. Optical rotation of an optically active compound depends upon two things. One is the concentration of the compound, and the second is the path length. The optical activity depends upon concentration of the compound, as well as it depends upon the path length of the light and the wavelength of the polarized light. Hence, there is a concept which is being given about the specific rotation. The specific rotation of any molecule. Now, what is this specific rotation of any molecule? The specific rotation refers to the optical activity of a molecule when there is a wave, then when there is the concentration of the solution is 1 gram per ml, and the length of the path through which the light is made to pass is 1 decimeter, then the optical rotation is equals to 1 angstrom. Okay, so optical rotation, the specific rotation is used to measure the optical rotation when the path length of the cuvette is 1 decimeter and the concentration of the solution is 1 gram per ml. Okay, now I would be speaking in Hindi also. If there are students cannot understand it, please just wait. This is all in English that I have to summarize this for some other students also. देखिए, if अगर आप कभी भी स्पेसिफिक रोटेशन को मेजर करना चाहते हैं, then you have to measure optical rotation in degree. One angstrom optical rotation in degree per 1 gram per ml concentration of solution and 1 deciliter decimeter length of the cuvette, you will get the specific rotation. specific rotation measure करता है भैया optical rotation को जब solution की concentration 1 gram per ml होती है. Specific rotation measures optical rotation when the concentration of the solution is 1 gram per ml. Okay. Now there is a question which was asked in CSIR, and it's an easy question. You just try to solve this particular question along with me. See, there is a solution. There is a solution which is being given. Okay, and the solution has a concentration present within itself. Now, what is that concentration that you can look forward? The concentration is 3 gram per 50 ml of 6NHCl. Okay, there is 6 normal HCl, and there is 3 gram of a solution of a substance taken in 50 ml. Can you calculate the concentration in gram per ml? 3 divided by 50 would be the concentration in gram per ml. Okay, has an observed rotation of, observed rotation means the value of alpha is being given, that is 1.81, and in a 20 centimeter polarimeter tube. When you are talking about polarimeter, this entire instrument that measures the optical activity of any molecule with respect to the light is referred to as the polarimeter. This instrument is referred to as the polarimeter. Now, this polarimeter can be effectively used to measure the optical rotation. Now, what we are going to put here is that 1.81 is the observed rotation. Okay, because this is a 20 centimeter polarimeter tube, and you have to measure the things in decimeter. Decimeter, 1 decimeter equals to 10 centimeter. The 20 centimeter will be equals to 2 centimeter. This is the length. Okay, and the concentration is 3 divided by 50, 0.06. So here it comes out to be the optical rotation at specific temperature, at specific density in a concentration, at a concentration of 1 gram per ml. That optical rotation is called as specific rotation. Okay, that optical rotation which is taking place at a concentration of 1 gram per ml at a standard temperature and at a specific density is called as specific rotation. Hopefully this would be understood to you. Okay. Now we are going to move forward. Such type of questions are given in your MCQ. You can solve those questions. Okay. Now there are two different types of absolute configurations. Now what do you mean by absolute configuration? Configuration, as the word indicates, see this is a very important word. When you talk about a word that is called as configuration, then the word configuration itself says that there can't be a simple rotation. There can't be a simple rotation, that this molecule cannot get rotated into the other molecule. Okay, the rotation is not possible. This is not possible. The rotation in between these two molecules and their interconversion is not possible when it is written configuration. Whenever it is written conformation, see at this place, if there would have been conformation, that means it means that the two different molecules can get interconverted into one another. Okay, if there is a word conformation, this means just by rotation you change one molecule into another. Conformation means rotation. You just make a rotation and you would be able to convert one molecule into another. Okay, this is rotate one molecule into another is referred to as conformation. When you cannot alter the two molecules into one another, then it is called as configuration. In terms of layman language, अगर आपको कुछ नहीं याद रहेगा तब भी मैं याद करा दूंगा. Even if you cannot remember anything, then also I will make you remember everything. For example, if the forms are being written in the format of capital L or capital D, always remember this is absolute configuration. In capital L and capital D, the L-glyceraldehyde can never get converted into capital D-glyceraldehyde. But if this L and D will be written in small letters, then it is conformation. Okay, and there the L will get converted into D and D will get converted into L in small letters. Where it is being written, just it is coming at the towards the end of this particular page. That is referred to as conformation. This is referred to as configuration. For example, glycerol D has two enantiomers. Now, what do you mean by enantiomers? Enantiomers are mirror images to each other. Supposingly, you keep a mirror in between these two structures, then the two structures will be absolutely mirror images to each other. But this structure, if you're going to take this structure, put it over here, this would be non-superimposable. Mirror images that are non-superimposable are said to be enantiomers. Okay, in the prefix D and L refers to the absolute configuration of the glyceraldehyde. Similarly, absolute configuration of the amino acids are of two different types, D and L amino acids. We always have L amino acids and we always have D sugars. Human beings and the living world always, usually the eukaryotes have L amino acids and D sugars. Eukaryotic organisms have D amino acid, have D sugars and L amino acid and L amino acids. Okay, so this is very important. You must remember this out. Okay, this has been asked some several examinations here and there. Okay, now moving forward. Okay, you get all these different types of configurations that are possible. Now, which is the only amino acid that will not exhibit the D and L conformation is the glycine, because glycine is not going to have the side group of methyl group or any other group. So glycine will not be able to exhibit any absolute configuration. Okay, now, moving forward, there are other different configurations which are also possible. One is referred to as the R configuration, another one is referred to as the S configuration. R configuration refers to the rectus and S configuration refers to the sinistral form. Now, what is this sinistral and what is this restral form? Now, this refers to the rotation of the plane polarized light along the chiral carbon atom, in which direction it is moving. So you can find that in the case of L-glyceraldehyde, it is moving in left-handed direction. In the case of D-glyceraldehyde, it is moving in right-handed direction. Okay. So R is assigned the configuration. See, this is configuration. This is not conformation. This is just the rotation of the plane polarized light. So this is configuration. Okay. R is the configuration where you can look here. Configuration is assigned by looking down the bond to the lowest priority, that is to the lowest molecular weight. As you move from the highest molecular weight to the lowest molecular weight, if you are moving in the clockwise direction, you give the term that is referred to as R, and if you are moving in anti-clockwise direction, you give the term that is referred to as S. Okay. Now, so the amino acids can have two different conformations here. One is referred to as the configurations. One is referred to as the R configuration, and another one is referred to as the S configuration. Now, when you are going to classify all the amino acids, all amino acids will exhibit the R form of configuration, except the one that is referred to as serine, that will exhibit the S form of S configuration. There was a problem with my connection. Okay, so majority of the amino acid will exhibit the R configuration, while some such as the serine exhibit the S configuration. Okay, Now moving forward, what you are going to get is that about the information about the standard amino acid. Although there are more than 300 amino acid known, but in the case of cells, there are only 22 standard amino acids which are present. What do you mean by standard amino acid? Now, standard amino acids are the one that are being incorporated during the protein synthesis into the structure of protein. When the amino acids are incorporated in the structure of protein during its biosynthesis, then all such amino acids are referred to as the standard amino acids in nature. Okay. Now, what we are going to look here is that the standard amino acids which are being given here are the 20 different types of standard amino acids that you can find. You must remember their triplet letter and their singlet letter. There is no trick to remember the triplet or the singlet letters. You will have to mug them up, or you will have to just cram them up. For example, glycine has GLYG. It has the lowest molecular weight, the simplest type of all amino acid residue. I've already told you: alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine. See, the amino acid with the maximum occurrence in protein, because it has six number of synonymous codons. It has six number of synonymous codons coding for itself. That is leucine. Okay, the protein, the amino acid which is minimally found in protein is tryptophan. Maximum is leucine. Tryptophan is minimum. Tryptophan has the maximum weight. Okay, and the minimum weight is that of glycine. Weight based questions have also been asked in other competitive examination, including CSI, and those are given in your book, in the MCQ book. Okay. Now, amino acids can be classified on the basis of the charge that is present on their side chain. Now, see this: amino acids which are non-polar side chain are glycine, alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine, and tryptophan. Whereas charged polar side chain means that there is a side chain which contains some charge present over itself. As it contains certain charge present over itself, that is why it is referred to as a polar side chain containing amino acid. You can look towards the structure of serine. Serine will have a hydroxy group. Threonine has an hydroxy group. Cysteine has a thiol group, SH group. Asparagine has an CONH2 group. Glutamine has an CONH2 group. Tyrosine has an hydroxy group. As they are all having an electronegative element containing side group, so they are all polar side chain containing amino acids. क्योंकि इनके पास में क्या है, साइड चेन पे क्या लगा हुआ है, इलेक्ट्रोनेगेटिव एलिमेंट कंटेनिंग amino electronegative element containing groups are being added in their side chain. That is why all of them are uncharged polar side chain containing amino acids. Amino acids with charged polar side chain. Now, which are those amino acids with charged polar side chain? All the basic and the acidic amino acid. Acidic are aspartate and glutamate with the minimum isoelectric point of 2.77 and the maximum that of arginine. Arginine structure contain guanidino group. I will be speaking to you about that also. Lysine, arginine, and histidine, RKH. You can remember it by the term RKH. Okay, RKH can be used to remember this, and here it is DE that could be used to remember aspartate and glutamate. So you must remember their names at least. Okay, then we are going to start in tomorrow's class about the structure of different types of amino acid residue. You have to practice these structures thoroughly so that you can draw these structures. I would be making a trick and I would be providing a classification so that you can remember the structure of amino acid residue much effectively. Today class will be shared with you on the YouTube as well as other media. Okay, so tomorrow I'm going to take a elongated class, take a long class from 7:30 to 9:30, and there would be a 10 minutes break in between that. Okay, so that's all for today. Today I'm closing here only because you have to learn all the different names of these amino acid residues, as well as you have to memorize the structure. Try to memorize the structure. See, look at the structure of glycine. It is the simplest of all. There are two different protons which are present. Then it is glycine. Look at the structure of alanine. Just a methyl group has been added. Which has been added? A methyl group has been added. You just add a methyl group to the structure of glycine and you make another structure, my dear friends, and that is alanine. See the structure of valine. The structure of valine becomes clear from its first letter, that is written as V. You just look here, invert V and add it here. Okay, just invert V and add it here. Then what you get? Just V could be inverted here and you get the structure of valine. Now, what is this leucine? In the structure of leucine, there is one more carbon atom, and because it is containing L, so this part has been elongated. Okay, this part has been elongated. Elongated because it has an L present within its structure. If you can remember leucine, leucine can be isomerized into another molecule that is referred to as isoleucine. Now, leucine has this methyl group. You transfer this methyl group to the beta carbon atom. When you transfer it to the beta carbon atom, you form isoleucine. Okay, you form a structure of isoleucine. Okay, now the next thing is that about proline. You can remember the structure of proline with the help of a temple. This is temple with a window. Okay, this is a temple which has a particular window. And where is the window present in our temple? Window is always present at the towards the top in the temple. And see this, this is the window that is present in the temple. You see this, this is the structure of the temple. And what you find here is that here is the at the carboxylic group, you can find the flag of the temple flying away. This is the flag. This is the flag of the temple that is flying away. Okay, you can look at here. This is the flag of the temple that is flying away. Okay. Okay. So this is the structure of proline. It is cyclic in nature, but it is not aromatic. Okay. Methionine, it has a effective group, thiol group present within itself. Although there is an effective thiol group, my dear friend, present here, but this thiol group is never going to form a disulfide bond. Okay, it has a thiol group, but it can never form a disulfide bond. The aromatic amino acid are phenylalanine, tryptophan, and the third one that is referred to as tyrosine. Okay, there are three only amino acids which are aromatic: phenylalanine, tyrosine, and phenylalanine, tyrosine, and tryptophan. Okay, just clearing all the drawing. Moving forward. Now, how to remember this tryptophan, because this has a very complex structure. You just remember this tryptophan as CCHNH. Once you remember this word CCHNH, just invert it for histidine. What you will get it for histidine, please do not annotate. Okay. In the case of histidine, what you are going to get, where is histidine? Okay, there it is. Cannot find histidine somewhere. Arginine, lysine, histidine will be given as such. Ooh, this is histidine. CCHNH. At the place of CCHNH in tryptophan, what is given here? CNHCH. See this. You compare this tryptophan with this histidine. Okay, you will be getting CCHNH there. CCHNH. Here, what you are getting? CNHCH. So you can find that both these molecules differs in the orientation of this high NH group. NH group is present at the third position in the case of tryptophan. In the case of histidine, it is present at the second position. And then you again repeat N and C, and you get a ring-shaped structure. But this histidine is also not aromatic because it do not contain alternate double bond present within its structure. Okay. Now, after tryptophan, there is the serine which is present. The serine could be effectively remembered as the hydroxy group containing amino acid. Then there is threonine. You can remember threonine via a trick. Threonine always confuses. OH is ordered not at the second carbon atom but at the first. Threonine confuse करता है, OH दूसरे पे नहीं, OH तो पहले पे लगता है. Okay, threonine always confuses. OH is never added on the second carbon atom, it is always added on the first one. Okay, then cysteine containing the thiol group. Arginine, well, arginine and the aspartate, there would be no difference. The only difference will be that of the NH2 group that is present here. Glutamine and glutamate, there would be no difference. The only difference, there is no carboxyl group and there is an NH2 group present here. Okay, tyrosine and phenylalanine, the only difference is that of the hydroxy group that has been added over the phenylalanine. It will lead to the formation of tyrosine. Okay, there are three positively charged amino acid residue. This group, which is present in the case of arginine, is referred to as the guanidino group. Very important group. This group is referred to as the guanidino group. This group is commonly called as the guanidino group. Okay, this is the guanidino group that is present. Lysine, it is the amino acid that has the elongated carbon chain present within itself. Okay, then there is histidine. In the case of histidine, what you are going to find, this is going to be present at the active site of all the enzymes because it has an isoelectric point of 6.8. Okay, and then there are two negatively charged amino acid residue. Those are that of aspartate and glutamate. You can find there is an one less carbon atom. There is only an alpha and beta carbon atom, but there you will find alpha, beta, and gamma carbon atom. Gamma glutamate will be present. So I'm just winding up my class here only for today because there are so many things that you have to learn about the structures of different amino acids. Please make it sure that you can learn about these structures because these structures will form the basis of the biochemistry that we are going to learn in tomorrow's lecture. Okay, tomorrow you will also have a test. Please try to give your text in between 5 to 7:30, and from 7:30 you will have a regular class up till 9:30. Okay, thank you, thanks a lot. Hope you enjoyed the lecture. You will get the recording of the lecture on our official YouTube channel. The link of that will be shared with you in the group where you will get it. Okay, okay, thank you, thanks a lot. Tomorrow, like tomorrow class also, the lecture has not been uploaded to YouTube because YouTube is creating some trouble for very long lectures, but we are uploading it today by itself. Thank you, thanks a lot. All of you can move out now. Thank you.